Biological basis of memory. Pavlov believed that the memory involved in learning a classically conditioned response would be explained as a matter of changes in the brain. In the 1920s, American physiological psychologist Carl Lashley began the search for the memory trace, or engram, the brain changes associated with the formation of a new long-term memory. He believed that memory was localized. A particular memory is stored in a specific brain area. Lashley systematically removed different sections of the cortex in rats trained to run a maze, but he failed to find a specific location for the memory. At the end of his career, Lashley concluded that memories are not localized in specific locations, but instead are distributed or stored throughout the brain. This assumption was not entirely correct either. American psychologist and neuroscientist Richard F. Thompson confirmed Pavlov's belief that some memories are localized or stored in specific areas of the brain. He found that in rabbits, a classically conditioned eye blink response was formed and stored in a small area of the cerebellum. Combining the findings of Lashley and Thompson suggests that memories have the potential to be both localized and distributed. Very simple memories may be localized in a specific area, whereas more complex memories seem to be distributed throughout the brain. The notion of a memory trace suggests that some change must occur in the workings of the brain when a new long-term memory is stored. Two possible changes could occur. The functioning of the neurons could change. The structure of the neurons could change. In the last 30 years, memory researchers have studied aplasia, a sea snail with only about 20,000 good-sized neurons. Eric Kandel has studied the neuronal changes that occur when aplasia forms a new memory for a simple, classically conditioned response. The function of the neurons is altered. There is an increase in the amount of the neurotransmitters produced by the neurons. The structure of the snail's neurons also changes. The number of interconnecting branches between the neurons increases, as do the number of synapses or communication points on each branch. Collectively, these changes are called long-term potentiation, which refers to long-lasting increase in synaptic strength. Forming a memory seems to produce distinct functional and structural changes in specific neurons. These changes create a memory circuit. As the changes in the neurons strengthen the communication links in the circuit, the memory becomes established as a long-term memory. Amnesia is severe memory loss. Retrograde amnesia is loss of memory, especially for episodic information. Retrograde means backward moving. Memory consolidation is the gradual physical process of converting new long-term memories to stable, enduring memory codes. If memory consolidation is disrupted before the process is complete, memories of the events that immediately preceded the brain trauma are completely lost. Anterograde amnesia is the inability to form new memories. Anterograde means forward moving. Brain structures involved in memory. The cerebellum is involved in the classical conditioning of simple reflexes and in procedural and other motor skill memories. The amygdala seems to encode the emotional qualities associated with particular memories, such as fear or anger, as well as memories of sensory stimuli that are associated with rewards and punishments. The frontal lobes are involved in retrieving and organizing information associated with autobiographical and episodic memories. The prefrontal cortex seems to play an important role in memory, especially in the ability to remember the order of information. The medial temporal lobes are involved in encoding complex memories by forming links among the information stored in multiple brain regions. Dementia is characterized by decline and impairment of memory, reasoning, language, and other cognitive functions. Alzheimer's disease is the most common cause of dementia. The brains of AD patients develop an abundance of two abnormal structures, beta amyloid plaques and neurofibrillary tangles. Alzheimer's disease progressively destroys neurons in the brain. The disease first attacks the temporal lobes, affecting areas involved in memory. Next affected are the frontal areas, which are involved in thinking, reasoning, self-control. There is also significant loss in the limbic areas involved in regulating emotion.